So this will actually be not one-dimensional, it will be multi. And the work that I'm presenting here, the first half has been published last. The second half is work, which is submitted to us. If you guys have some feedback on, let's see. So what I'm saying is work of a whole team. From Mount School of Medicine in New York and his grad student both at Princeton, and the computational analysis was done at my lab. Olga is at Princeton, Avi is at my lab. Was by different labs, including Tony Wett, Richard Sterboy, who I think was or still is at the MIT. In right here is a lot more competition people, and I think that's a game. It's kind of easy to check the data sets, but then figure out what it all means. So to show how it looks like, let me give you a little cartoon here. What we work with is mouse embryonic stem cells. So these are cells which are pluripotent and they self-renew. If you just leave them in the right medium, just stay stem cells. They don't do anything. So that's what you see here. However, if you perturb them in certain ways, you kind of make them commit to certain cell lineages. And the perturbation we used is to knock down NANOC, which is one of the key transcription factors in mouse embryonic stem cells. It's one of the main gatekeepers that keep stem cells as stem cells. And what we did is, in day 0, we have the stem cells, then we knock down NANOC with an RNAi construct, and we follow the cells over 5 days. And at days 1, 3, and 5, after the perturbation, we take samples and we analyze them. And what we do at each of these 3 days is we look at 4 different levels in cells. 3 of the data sets are data sets. The first is a chip 9 and 14. The second data chip, it's where we look at global gene expression measurements on 2,000 new proteins made by mass spec. You see, that's like all very important. Even mathematician I am, will tell you acetylation is somehow it's get a lot of acetylation in the chromatin wheels. The polymers come in, bind to DNA, means then the first step we should increase in, of course, lead to an increase in that's my mathematician do. What we actually there's a lot of coherent levels. Did study we concentrate the genes which were not expression level, try to follow them back in the study. What you see on the protein level, the one always down here, a similar change. These groups that you see are which just is like, so that double check data, a lot more statistics. I think this is not, hopefully not, mechanisms of other things in stem cells. Postulation. The next thing we did is one dimension, right? So it was So we looked at how these expressions that so behaves. You see that network comes from a paper, script and target. And what you see here is at first they're highly expressed, and you can expression at day 5, they're all very low. After 5 days, are really no longer stem cells. Different. So we took a protein action data sets and we mapped our data on that. Little boxes is one, and in each box, protein expression for days 1 to 5. Years now, there was experimentally perturbed, so it's not a problem. But we on the bird's eye that those are ones that directly increase fast. They don't get signal fast that nano was depleted. How spread starting nano spreads through the network. One of the spin-offs of our paper at the lab of Avian visualize such multidimensional white, but what you see here genes are our for data of the little dots here. Little picture of how they look. You can click on the reason they develop. Compared to on mouse embryonic stem. About that is while other data cells, we have a stem cell state. Understanding the dynamics in the what we found is but the state if you have interest you can develop as this data set the protein level. What I looked at is expression and tried to profile of to get one interested in is using the for sequencing. So the probes were move that into different bits. 14 actually. And also have a description profile and correlation between those acetylation up and down. But focus. Maybe I find something in. So let's get the matrix, if you look at change on days 1, Pearson correlation, you see is actually 0.3 is very significant. In here is that over time. So small at the beginning, and things in clear patterns. This results in this correlation method matrix here. I hope what happens is pure correlation or level correlation changes doesn't go away. That's the clear pattern. Let's look at it. This is using classical statistical classification, and I looked at 4 particular classification problems. The first one is, can I predict whether genes move up versus whether it's move down, so transcriptionally, differentially up or down regulated, given their acetylation profiles? Can I predict whether they go down or don't change, up or don't change, or show any change up or down, or just stay stable? So it's 4 different problems. And they come with different data set sizes because obviously different number of genes that go up or down or stay stable. So to compare these 4 problems with each other, I used a resampling approach. I always took the smaller sample size and sampled from the bigger set. So I don't know, take the positive examples as the smaller set, sample from negative example, the set of the same size, such that the accuracy you would expect. That's just to make these different classification problems comparable. And then I used tenfold cross-validation to give a quantitative measure of how well the how big the predictive power is between acetylation and gene expression. And what you get is a little box plot like this, which tells you that, I don't know, the blue classification problem, which is any change versus no change, is basically just above the baseline, and classifying up versus no change is a bit better. The pink down versus no change is even better, and up versus down is really good. This is for one particular classifier. So I thought, maybe it depends on the classifier I use. So I just did it for all kinds of classifiers. So you see different flavors of the machine, or discriminant analysis, classification trees, k-nearest neighbors, all kinds of stuff that some patterns can see, which are very striking. Down regulation is always the simplest problem, because clear signals. The blue thing here, because there's a very interesting part here is that regulation. So and try to explain that events ourselves much larger acetylation events. Acetylation was acetylation happened on a whole wrong signal. So that's why it upregulated is much harder. The next thing I did is, and what I looked at is low profile again on they show low or high acetylation. What are the key gene embryonic stem cells? We know, and I have a different answer. So I thought, okay, let's just take off definitions of key genes genes, how they behave. And I came 5 different genes. The first one is called the Plurals on Nature paper from 2006 by Franz Müller. We use a computer, the machine learning method, to define a large gene spread compendium and protein interaction, and all the genes that end in his signature. From our for self. Third one is when I went to go and look up stem. The first one is the stem cell specific form of those slides. The last one are known transmarkers. What you see is a very striking pattern. Written off an gene you take, they usually are so they are very much made for high chromatin marks. And so one of the theories about what is they are wide for all the embryonic stem cell genes wait for transcription factors other regulators to come in. Now, chromatin be open, so the kind of is evidence for the hypothesis. Now, this is what happens over time. Look at the acetylation signal from versus day 0, so that's day, I guess, day 5 versus day 0. So some of them go up, most of them go down. It's the same 5 gene sets again, and you see now they're all on the left. Oh, by the way, so these dots here mean that they correspond to you. So 3 dots mean like really, really significant, and 2 dots mean not so much significant. 1 really not, just 10 to the minus 1 or something. And that was evaluated by gene set analysis, which directly measures whether you see a trend in a certain direction. So what you see here is significant trends now to to the deacetylated side. So the interpretation of the last plot on this plot, that while the key stem cell genes were very widely open in the beginning, as soon as the genes started to differentiate, they are rapidly closing down. So these plots compare the cell genes of all the other genes. So if they're on the left, means they're closing down faster than all the other genes in the gene. Now, is this reflected in the gene expression change? So it is with gene expression down or up, and I found almost nothing. So these guys here, they're just over the place. They're completely randomly spread out. The gene is closing down. On the description level, we have 4 description factors down here. That's kind of positive control, because these description factors are all known stem cell might go down description side. What is the reason that, for example, these large any trend to go down? And I think the reason is they are very specific. They are specific stem cell markers. You do an analysis of large compendium, and you put some specific that looks specific. You also specific genes, and the fluorine was especially designed to use protein genes that look important. And I think what this shows is also non-causal, non-important. Same holds true for the RNA. So the phenotype they use, which is, if you just find many non-stem cell influence on such function, and what we see here in this screen is just spread out, don't show the same kind of behavior genes like the stem cell markers do. If that is since we had the data presented on different embryonic stem cell incoherence between these different levels, the impact through transcriptional protein interaction data is the wealth of opportunity. One example I showed is where I express, and I could see is there's an immense, especially a very high acetylation beginning, which I want to thank for coming in.
Thank you very much. We have some minutes for questions.
So, you know Anog. After
Yeah.
5 heterogeneous cells. So how valid is it to look at experiments given working with homogeneous cell population?
I mean, we have the soup. I don't know this project, but if you look at here, it's just they did not select at any point for one specific lineage. They say it's just hard to do that. I think if they wanted to drift it forward, that's what they would have to do. Look for one specific lineage. I'm not sure if that's the reason they don't find this coherence. Actually, I think it should just average out. There should be more coherence than less. So that's kind of striking.
Okay, thank you.
Any other questions? I think a lot of questions, it's only that.
Yeah, I talked them into submission.
Yes. Other? I'm very fascinated about these things. I must say that one aspect that is never discussed in all these kind of experiments is something that was kind of touched before, but at another level, and it's a three-dimensional structure of all this. Because I remember a very classic experiment many years ago when there was a group trying to get expression of mammary gland cells, specific proteins, and they put any kind of factor that they could find, and they never got any expression of those genes. And suddenly someone came with the idea to grow this in collagen and then put it this as a ball, and suddenly the cells have three-dimensional contact, apical, cervical, and they will start producing these specific proteins. So I guess that many of the controls of these are not only at these levels, because where we were talking about DNA, again talking about one-dimensional cell monolayers. So that's maybe the reason that you see in Korea.
Yeah, it's like missing. Though 4, there are many more just in the middle. Yeah. Thanks again.
Thank you very much.